Okay. Hello, everyone. For those that are joining us, thank you so much. We're about to get started, and thanks for joining on to our webinar, Managing Potential Diseases in Vegetable Seed Crops. My name is Abir Day, and I am the Regional Coordinator in Ontario for the Bauta Family Initiative on Canadian Seed Security. As many of you know from our past webinars or from working with us in the field or through other engagements, ours is a program dedicated to increasing the quality, quantity, and diversity of ecological seed grown across Canada. And a big part of our work is delivering training to seed producers, and these webinars are an integral part of that because they allow us to bring together, folks from sometimes isolated seed communities and also share the knowledge of experts and practitioners across the country and the continent in a really effective way. So thanks for being here, and it means a lot that you're giving us your time to learn today. I just have a few administrative things to get out of the way before we get started. This is the third webinar in our new webinar series. It's called Seed Systems, Broadening Our Knowledge. Sorry, I just have to stop right here. Just if there's anybody who's joining by phone, be sure to mute your line by pressing *6, and to mute your speakers and any microphones on your computer. Otherwise, we get the echo that we would have just heard. So this is the third webinar in our webinar series. We're excited to be using this kind of platform to explore a range of topics that go beyond seed production and into broader aspects of our seed system. And for those that are looking for more beginner-level seed-saving webinars, we're also partnering with the Heirloom Seed Sanctuary in Kingston, Ontario, with Kate Henderson, who's leading an excellent beginner-level webinar series. And you can find that out on the Heirloom Seed Sanctuary's website. Our full webinar schedule is posted online at seedsecurity.ca. Those are the links that you're seeing right in front of you. And we have videos from all of our past webinars, and those links are on your screen right there as well. The last webinar of the year is going to be a little different than what we've been doing in the past. It'll happen sometime in the fall, and we'll be looking to you for ideas about what issues we should be exploring and what presenters we should be looking to engage with. So if you have any ideas for us, email us anytime at seed@acorn.org, acornorganic.org. And you'll have an opportunity to give suggestions when you register for any of the other webinars that are happening throughout the year. So we'll have a look at what themes and ideas come out of those suggestions, and we'll design a webinar around them. So stay tuned for that. Once again, if you're having any feedback on your computer, or if you can't hear us, check your computer and speaker settings, make sure they're on, but mute your microphone so you don't get any feedback or volume. And if you are having trouble getting audio turned on through your computer, you can dial in using the conference line that's posted above, but please mute your line on *6 by pressing *6. Okay, and today we'll be hearing from Lindsey du Toit about seed diseases. We're switching up the format this time, and Lindsey is inviting you to write your questions and comments throughout her presentation, and she'll address them as she goes, as she's able. And you can do this using the chat pod that's at the bottom right corner of your screen. Lindsey's prepared a very in-depth presentation for us, so please do let us know as you have any questions, and we'll pause frequently to address them rather than doing a dedicated Q&A at the end of the webinar. But if we find that we're just getting too many, we'll put a few in the parking lot, and we'll make sure that we can address them after Lindsey's completed her talk. So without further ado, I'll just give a quick bio for Lindsey, who's on the screen right now. And she has a really storied sort of biography, but so I'll do my best to kind of abridge it and still do it justice. She is a professor and extension specialist in the Department of Plant Pathology at Washington State University. Lindsey specializes in vegetable pathology, in particular vegetable seed crop pathology. The focus of her applied research and extension program is the etiology, biology, and management of diseases that affect vegetable seed crops grown in the Pacific Northwest, U.S., in particular small-seeded vegetable crops such as spinach, brassicas, carrot, onion, radish, and table beets. She's received a number of awards from several different institutions, societies, and organizations over her well-decorated career in plant pathology, and she co-leads the Pacific Northwest Vegetable Extension Group, a tri-state team of interdisciplinary vegetable specialists. We're really lucky to have her today, and it sounds like we'll need a webinar that'll last a few years to learn what she knows, but I think this hour that we have with her is going to be a great start. So Lindsey, take it away when you're ready.
Thank you very much, and I hope we can do some justice to the topic here. It's always difficult being asked to condense the massive field that you study into a short webinar, but hopefully this will give you some information that's of value to you and also help you recognize perhaps some avenues that you might need to explore. So I originally was asked to talk about potential diseases in seed crops, and the group agreed to kindly let me focus on vegetable seed crops because that is the area in which I've focused for the last 15 years, and therefore primary use of examples from vegetable seed crops to illustrate the principles we'll be talking about today. So I'd like to start off by quickly encapsulating why we worry about diseases in seed crops. Most of you think, well, this is obvious, but sometimes there's some aspects of diseases in seed crops that we really might forget and really have a tremendous responsibility to remember if we're involved in seed crop production and distribution of seed. And the most obvious, of course, is this disease might affect the yield of your seed. And that can be both the amount of seed, so the quantity in terms of yield, but also the quality of that yield. It may reduce, for example, the third bullet point in there is seed germination and seed vigor. Besides the direct yield loss, if the seed can become infected with that pathogen, excuse me, then of course that's a very big concern. If you're distributing that seed, you potentially become the typhoid Mary or the typhoid John if you are a source of infected seed. And some of these pathogens can also reduce the quality of the seed in terms of germination and vigor and shelf life, which of course is not good if you need to hold on to seed for a while, or if you have amount of seed that you'll be distributing for a period. And then of course what is very important is if you're distributing the seed, there's the potential to disseminate the pathogens. And even if it's the pathogen that perhaps is already established in an area, you may be introducing new races, excuse me, or new strains of the pathogen to an area where perhaps they've got old strains that aren't particularly virulent, and maybe there's good resistance, but if you introduce new races that can overcome that resistance, you can exacerbate an existing disease as well. Okay. Oh, Dwayne Fall commented that his onions look just like the picture on the cover slide. Well, I'll quickly go back, and for those of you who want to know what this picture is, this is a picture of botrytis infection in an onion seed crop, and it can infect the scape, the seed stalk, and it can cause blighting of the umbel. And we can talk a little bit about that later on. So I'm sorry, Dwayne, that your onions can look like this, but there's some really good things we could discuss about that perhaps later. So we tend to look at management of diseases in seed crops into three broad categories, and anytime you start categorizing stuff, there's going to be exceptions and exemptions to these rules that we try to create as humans. But we tend to focus on how do we manage these diseases culturally, what kind of cultural practices can we implement, and this becomes particularly important in organic seed crop management, as well as chemical applications and seed treatments. And this is not just reserved for the conventional side of vegetable seed crops, but also organic, because there are organically approved or certified treatments that you can put on. And even with seed, things like hot water treatment or certain disinfectants on seed can be approved under the organic certification programs. Excuse me. And lastly, we have this category of disease resistance. And this becomes very difficult because sometimes there may be resistant cultivars available for certain diseases, but as a seed grower, you may be dealing with the need to produce a certain variety, open pollinated or whatever type it might be, that happens to be very susceptible. And that can be really difficult. Not only do you potentially have susceptible variety, but you also typically in a seed crop have a much longer season, which means a much longer potential risk for things like diseases to cause problems. And before we get into the minutiae of these management practices, I always try to include some version of this pathogen life cycle in presentations I do that discuss the basics of diseases, because we tend to often forget what it takes for a pathogen to establish in a crop and spread and so on. Is, you know, the crop has to become inoculated somehow, and that can often be a very important question to ask yourself when you have a disease. Where does that pathogen come from? How did it survive? How was it introduced into your field? What conditions actually allowed that organism to infect and penetrate into the plant, establish and colonize and grow and reproduce and spread? And understanding the aspects of the life cycle of the pathogen, in this case, if we're specifically talking about pathogens, can be very, very helpful for understanding where your practices and how you grow your crop can perhaps be modified or not be modified to take advantage of what I call the Achilles heel of the pathogen. Different pathogens have different Achilles heels, and they may be completely different. Sometimes they're fairly similar. But recognizing the weakest point in the pathogen life cycle can often give you a leg up on trying to get better control of certain diseases. So just keep that in mind as you deal with the diseases that you might encounter in your area. So if we get back to that first broad category of cultural practices, this is a long list of the kinds of things that people talk about when you mention cultural ways of managing diseases. And I'll be giving some examples to illustrate some of these. Obviously, one of the most obvious is crop rotation. Don't plant the same crop on the same piece of ground over and over again, and that everyone tends to know that. But it gets more complicated, and we'll talk a bit about crop rotation. What kinds of diseases does crop rotation work better for than others? Another one is eliminating alternative hosts, and that means understanding what other hosts besides the crop you're growing potentially can allow that pathogen to keep reproducing and surviving in your area. The third one is destruction of inoculum in the field, and we'll talk a little bit about some examples of this. Maybe incorporating residues at the end after harvest into the soil if it's not a true soil-borne pathogen. There's the old practice of burning residues, which is controversial in itself and not always allowed. There's ways of modifying your irrigation practices to minimize disease pressure. Same thing with planting practices. And we'll talk about all of these. I'm just giving you the quick summary list, and then we'll go into some details with examples. Another really valuable one is ventilation of seed crops. A very dense canopy, tight row spacing, opposite direction to the main wind of your rows. All of this can compound and exacerbate diseases or be utilized to help reduce disease pressure. Fertility, the type of fertilizer you use, the amount of fertilizer, the timing of the fertilizer application. Some diseases can be significantly improved in terms of disease management by modifying your fertility program. We'll talk a bit about how practices of transplanting can impact disease pressure, as well as harvesting practices. And then one of the sort of more big picture perspectives is the geographical regional location where seed crop production is done. There's a very big reason the majority of the vegetable seed crop production, and many, not just vegetables, other crops, seed production is done in the western parts of North America, where it's drier and less conducive to diseases like fungal pathogens and bacterial pathogens. So this is the big picture perspective on what we mean when we talk about cultural practices. So let's go into a few examples here. So with crop rotation, this is where it's important to understand what species of plants are susceptible to the particular diseases you're concerned about. Some plants may be non-hosts, where there's no risk at all of that crop becoming infected. And those, of course, would be your ideal crops to rotate to when you're trying to manage a particular disease. Or you may have resistant cultivars, varieties that you know just don't get that disease or get very, very little of that disease. That can be helpful in the rotation. And then we have the so-called antagonistic crops. Some good examples are, for example, some of these biofumigant crops, like the high glucosinolate mustard cover crops. Arugula is often used in this capacity. These are crops that actually antagonize the pathogen in some form or another. In the case of the brassicas, these mustards that are grown and incorporated, or the arugulas and so on, they release these natural biofumigant compounds that help kill off inoculum in the soil. And they can be very valuable for reducing disease pressure, particularly the soil-borne pathogens, which can be much harder to manage than the foliar pathogens. A big question that often I get from growers is, how long do I have to stay out of this field with this particular crop to avoid this disease showing up again? there's a lot of things that influence that. How long do I have to rotate out of the field? That's going to depend on how wide a host range the pathogen has. And pathogens, many of them are very specific in the types of species they will infect or one particular species they'll infect. Unfortunately, there are some of those notorious pathogens like Sclerotinia, which causes white mold, that have a very broad host range, and it becomes a much more difficult selection of rotation crops to avoid continuing to increase the inoculum in your soil. And so that's why it's important to understand the host range of your crop, the disease you're working with or you're concerned about. Another important factor on crop rotation is whether the pathogen that causes that disease is what we'd like to call in pathology a truly foliar pathogen. It causes losses above ground, it primarily exists above ground, or if it's a soil-borne pathogen, it has the capacity to persist in the soil for an extended period. Foliar pathogens, like many of the leaf spots, they do not survive very long in the soil in the absence of host residue. So crop rotation is very effective against diseases that are foliar and can be much more difficult to work with on soil-borne diseases. And we have some examples of diseases where growers have to stay out of a field for at least 10 years if they know they've had a certain soil-borne pathogen, and I'll give you some examples of that. So this kind of ties into the next point here about the longevity of inoculum survival. Some pathogens do not persist very long in the soil. They'll only persist in the soil as long as there are residues, crop residues from after harvest. Some pathogens don't care if there's crop residues or not, and a very notorious one for those of you who do garlic is white rot. That white rot inoculum can survive in a field 20 years without any garlic or any allium species, and as soon as you plant an allium species, it will start to germinate the sclerotia, and then lo and behold, you have white rot even though you haven't had an allium crop in that field for 20 years, and that's one of the more notorious examples. It also depends on the level of resistance or cultivar of the cultivar or the parent lines that you are growing. And of course, it depends on things like cultural practices. They all sort of tie in as to how long inoculum might persist in that area. One of the things that's really important to be aware of in this topic of how long you have to stay out of a particular species in the field is whether or not there are any asymptomatic hosts for that particular disease. This can be problematic for things like certain viruses. Asymptomatic hosts are plant species that can become infected but don't show symptoms very well, and you may not recognize that there's these plants out there, whether they be weeds or whether they be another crop you're growing that perhaps can keep the inoculum level up for that particular pathogen. So when you go back to the very susceptible crop, you haven't really gotten rid of the problem. Another factor to consider in crop rotation is the alternative hosts for that pathogen that might be present in your area. Weeds can be very notorious for this. Those of you who grow brassica species, you may have heard of the disease blackleg caused by Phoma lingam. And unfortunately, almost any plant in the brassica family or the crucifer family has the potential to be infected. And we have many, many brassica weeds in most parts of the world where agriculture takes place, and they can be a notorious alternative host. Volunteers, this can be a big issue when you harvest the crop, and particularly with seed crops, you have some of the seed that shatters and falls to the ground and grow through the winter or perhaps the next season and produce these volunteers. And if that seed was infected or there was inoculum still present in the field, they can continue to propagate that pathogen. And adjacent crops, crops that are perhaps on your neighbor's farm or your farm but in another field that perhaps have inoculum, especially for pathogens that can spread distances by wind-blown spores and so on. That can be a problem if you're concerned about where inoculum might be coming from, is recognizing how adjacent crops can sometimes provide a source of inoculum. One of the topics, I'll show you some nice illustrations, diseases to illustrate this, is the concern about overlapping biennial and annual crops. Some of the vegetable species I work with are grown in the conventional crop as an annual, for example, carrots and onions. But for seed crops, they're biennial. And now you have a biennial crop sometimes grown in the same area as an annual version of that crop, and it creates what we call in plant pathology a green bridge. There's always host material present year-round. So if the pathogen is present, there's always something to infect, or if For example, it's an insect-vectored virus or an insect-vectored bacterium. That insect can just move around and constantly have a host to feed on. So that green bridge can be really, really difficult for disease management if you are in an area where you have these overlapping seasons. Okay, doesn't look like we have any questions, so I'll move on. Here's a classic example that illustrates some of these points we've just discussed. So for those of you who work in allium crops, you might have heard about a pretty notorious virus that's showed up about 20, well, I guess now 30 years ago in the U.S. in Idaho, and kind of didn't do very much for about 10 years, and then in the 90s started spreading like wildfire and is now found almost anywhere onions are grown in the U.S. and certainly almost anywhere in the world now. Iris yellow spot virus is carried by thrips, a tiny insect that the adults can fly and move around, and it can infect almost any allium crop. Some alliums are less susceptible, but onions are very, very susceptible. And here are some photos, 3 different photos, showing the range and symptoms that you can get in an onion seed crop. The picture on the right shows why this disease can be quite problematic in onion seed production. These lesions on the seed stalk or scape can get quite large. They can girdle the seed stalk. And when the umbel starts to fill, that seed is filling, it gets heavy. And if you have any kind of wind, those seed stalks collapse and fall over. And not only does this reduce the movement of photosynthase into that developing seed, so you get smaller seed and you get less seed. If your seed crop is machine harvested, those heads don't get picked up. If they're hand harvested, you can recover a tremendous amount of that. But for machine harvesting, these heads are a loss. So there's a picture of a little critter that vectors the Iris yellow spot virus, the actual pathogen. Not only do these thrips feed on onions themselves and can potentially cause some damage, but they can carry this virus. And I'm going to show you some pictures and illustrate some points about this movement of the virus on the thrips and survival and this green bridge effect. Here in the Pacific Northwest, we have a lot of hosts, annual and biennial, of different allium species that this virus can persist on. We have volunteers that grow as a result of plants left after harvest of bulb crops or shattered seed from onion seed crops. Sometimes transplants. We can have some asymptomatic hosts. There's evidence that quite a few weeds can become infected with Iris yellow spot virus and not show symptoms. Then, of course, thrips themselves can move around with onion bulbs. When you perhaps are moving bulbs from one area to another, selling bulbs, it's not uncommon to be able to pull back the scales and find a few thrips on there. If they're carrying the virus, in other words, they're viruliferative, they could be spreading the virus. The one saving grace about this virus, which is amazing that there is one, is that it's not seed-borne, so you don't have to worry as a seed grower that you are selling infected seed. But the green bridge effect has really become a primary driving force behind the widespread distribution of this virus in the Pacific Northwest. The second picture I've just brought up on the screen shows an aerial photo of an onion seed crop. This is in Central Oregon. And the brownish light green area is very, very severely affected by Iris yellow spot. And you can see the darker green is off to the right in this photo. The bare ground field immediately to the left of this crop had an onion seed crop the previous season. And as that crop was maturing and nearing senescence, when the seed is drying, the thrips basically flew over to the field to the right, which is where the new onion seed crop had already been planted and had been growing for 2 months. So this green bridge effect just allowed the thrips to fly over to green tissue to feed and spread the virus with them. It's a classic example of a green bridge effect that we deal with, especially with insect-vectored viruses. This is another aerial photo of taken in central Washington. The big field that's sort of circular shaped on the left is an onion bulb crop. This is an aerial infrared photo, so red means healthy and brown means unhealthy. To the right in the center is a square field or a rectangular shaped field that was an onion seed crop. And about 2 weeks prior to harvest of this onion seed crop, a large percentage, about 80% of the scapes, the seed stalks fell over. This was the seed crop I visited with the seed grower. And we managed to get these aerial photos through the extension agent at the time, Mark Trent, and you can see the direction of movement, the gradient of movement of the thrips as they flew out of that drying dying onion seed crop into the onion bulb crop, and this would have been in July, you can see that strong gradient direction of movement of the thrips carrying the virus. So again, this is that green bridge effect, which has really created a problem where onion bulb growers and onion seed growers are in the same area. Okay, any questions about that aspect? I don't see any on the screen. Okay, so let's go on to the next aspect of culturally managing diseases in seed crops. And I mentioned that sometimes you can try to destroy inoculum in a field. So you're removing or reducing the amount of infected debris or the amount of seed remaining after harvest in an attempt to reduce inoculum, and this can be particularly important for soil-borne pathogens. Things like burning stubble or debris, I've mentioned that this has historically been a very valuable way of getting rid of inoculum, but doesn't always allow it because of other aspects of environmental contamination and risks and so on. I have heard about the practice of vacuuming fields. This may seem a bit ludicrous, but with things like ergot in grass seed production, the ergot sclerotia can actually be vacuumed up if you've got a small enough field, and you can buy these commercial vacuums that suck up the sclerotia, as well as a whole lot of other stuff, I'm sure. Another way of destroying inoculum is fumigating the soil. Now, this doesn't just mean using these hardcore, nasty, very toxic synthetic fumigants, but biofumigation is also an example. I mentioned these mustard cover crops that are grown very widely in the Pacific Northwest for various reasons, and one of those reasons is to, when you incorporate all that biomass, it releases these natural fumigant products, the glucosinolates, that get broken down into isothiocyanates. They're toxic to pathogen fungal spores and other things in the soil, and they help reduce the inoculum. They can be quite effective and very helpful in addition to introducing organic matter. For those of you who work in areas where it can get very hot and very sunny, soil solarization has been used fairly effectively, but you have to be in an area that has a lot of heat and a lot of sun, and unfortunately for people who are in areas like maritime Pacific Northwest, not particularly conducive to soil solarization, but this has been used. Okay, and I'm going to give you one illustration of a disease I've worked on in my job at Washington State University, where reducing inoculum has really can make a very big difference regionally. I work a lot with spinach seed production. It's a non-heat tolerant crop for seed production, so it's grown mostly in western Washington, western Oregon. And the two fungi that can cause leaf spots in spinach seed crops that are quite problematic in our area are Cladosporium variabile, which causes Cladosporium leaf spot, and Stymphalium botryoosum, which causes Stymphalium leaf spot. They're both foliar pathogens. They don't persist in the soil for a very long time. So here you can see the kinds of symptoms they can cause on spinach. And what we discovered in beginning to work with these pathogens is that both of these pathogens can survive through the winter in the mild maritime climate of the Pacific Northwest coastal area. Cladosporium, in the picture on the left, survives as, survives as, you can see this picture on the left, these small whitish spots on the leaves. These are volunteer spinach plants that grew after harvest of a seed crop because some of the seed shattered and fell to the ground. And spinach is very cold tolerant, so it grew through the winter. And these small spots on the leaves are a classic form of survival of Cladosporium variabile through the winter. We never were able to get Stymphalium out of these spots on the leaves. Every time we isolated, we found Cladosporium and occasionally Colototricum, another leaf spot pathogen of spinach. However, what we did notice is that in the picture on the right, you see these woody pieces of stem tissue, which is the stem tissue left after harvest of a spinach seed crop. Wherever these spinach seed residues were left on the surface of the soil, not buried, but left on the surface of the soil, these tiny pinhead-sized black structures that you can see on the slide here, these are the fruiting bodies of Stymphalium botryoosum that formed. And it actually goes through its sexual cycle of genetic recombination as it forms these fruiting bodies in the winter. If those residues were buried, these fruiting structures never formed. So it was really interesting to see that these fruiting structures necessitated exposure to air and light on the surface of the soil in order to form. And this next slide shows a close-up picture of those little fruiting bodies. I call them the Hershey's chocolate kisses of death because each of these looks like a little Hershey's chocolate kiss. Unfortunately, if you cut one of these open, you can see there's a little neck on it. If you cut it open, inside them are these large, well, for a fungus large, these microscopic sacs. You can see in the lower right picture circling one of those sacs called an ascus. And there are hundreds of thousands of these sacs lining this little black fruiting structure. Inside each of these sacs is 8 spores. And these spores here, you can see them on the lower left side individually after they've been released from that sac. These spores literally are shot out through the neck, through that opening of that fruiting structure, like little cannonballs, and picked up in the breeze and blown away. So if you don't bury the residues after harvest of a spinach seed crop, if you leave them sitting out on the soil surface, the fungus that's infected those residues will form these fruiting structures. It'll take all winter for those fruiting structures to mature and produce these spores. And in the spring, when it starts to warm up enough, and the temperature's right, the humidity's right, they will force these little cannonball-like spores out. They'll get blown in the wind, potentially blown to fields next door, and restart the cycle of infection again for the next season. And so, excuse me, I'll go back. What we discovered with this is it's very, very important in areas where spinach seed crops are grown. All of the farmers need to incorporate the crop residues after harvest of the seed, incorporate the residues that fall, not wait till the following spring to do that, because if they do, there will be a huge amount of inoculum increase through the winter. What the difficulty of this is that if 1 or 2 farmers or 90% of the farmers practice this, enforce this cultural practice of incorporating residues, but 10% do not, you're still going to have the problem, because that 10% who do not potentially are building an inoculum that can blow between and among fields. So this regional practice of destroying inoculum is very, very important. Other ways that we have looked at trying to help manage diseases through these kinds of practices. Here is a picture we took of some field trials we did looking at cover crops and soil amendments. This one was looking at the mustard biofumigant crops. You can see this is in the late fall. The mustard cover crop is flail mowed and then immediately incorporated to release those enzymes into the soil and not into the air, and to help control. In this case, we were looking at control of Fusarium wilt in spinach seed production. Another long series of studies we've done is looking at amending soil with limestone. In this picture in the bottom, I'm applying agricultural limestone, which is calcium carbonate. I think it actually came out of British Columbia. Applying it to the soil, because here in the western Pacific Northwest, maritime Pacific Northwest region, our soils naturally are very acidic with the high rainfall and the other materials that our soils are made of. pH is usually in the 5s, which is very, very conducive to Fusarium wilts. And so we found that by amending soil with limestone, applying it and incorporating it into the soil, we can raise our pH up as close to neutral as possible and very significantly reduce the amount of losses to Fusarium wilt in crops like spinach seed production. And here's a slide to illustrate this. This was a study we did in 2006 in a grower's field. We looked at limestone amendment at different rates from 0 to 4 tons per acre. And we grew 2 different female lines, one that was highly susceptible to Fusarium wilt, shown in blue, and one that was moderately susceptible, shown in black. Nothing is completely resistant in spinach to this disease. On the y-axis is seed yield in pounds per acre. And what you'll notice, first of all, let's look at the black bar, which is that female that has some partial resistance but not completely resistant. And you can see a very dramatic increase in seed yield as we increase the rate of limestone from no limestone to 2.8 tons per acre. And we went from about 1,300 pounds per acre to 2,500 pounds per acre, which is very, very dramatic. This is marketable seed afterwards that we cleaned and sized. But what you notice, too, is as we increased the rate of limestone further, there was a huge and very rapid decline in the seed yield. And one of the things we discovered is as we shift that soil pH up towards neutral, the availability of certain micronutrients in the soil decreases. And for seed crops, it's really important to have an ample supply of micronutrients like manganese and zinc. Otherwise, you can have a very adverse effect on seed yield. So there was an optimum rate to this practice of putting limestone on and not shifting the pH too far without supplementing the crop with a foliar feed of zinc and manganese. For the very susceptible female, it looks like you can see that as we increase the rate of limestone amendment, the seed yields continue to go up. Didn't look that too dramatic, but when you start with a seed yield that is that low because of how susceptible that female was, this actually represented a 300% increase in seed yield because of how severe the losses to Fusarium wilt on that crop. We had a PhD student who did a series of trials on this practice of limestone amendment. I won't go into detail on this, but if anyone would like more information, they're welcome to contact me. And Emily looked at whether growers do no limestone application over a 4-year period from 2009 to 2012, or put on 1 ton of limestone every year for 4 years, or 2 tons of limestone. And you can see again the y-axis is seed yield. And you can see the dramatic, very dramatic increase in seed yield as a result of putting on an annual application of 1 ton of limestone versus putting on a single application of 2 tons after 4 years. Just trying to optimize the way we can use this and make it economically viable for growers. So, any questions from anyone regarding some of these practices of reducing inoculums? Okay, so let's talk a bit about, okay, someone is busy typing. I'll wait to see Melanie, Stacy, and Elena. Okay, so the question that Melanie, Stacy, and Elena asked is the increase in yield mainly related to better production or only reduction in loss from disease? It's a very good point, Melanie, Stacy, and Elena, and I, because of the sake of time, I didn't go into the details, but certainly it is a combination of multiple things going on. I didn't give you all the information. It was a very long and detailed study, and you're welcome to, if you want to read Emily's PhD dissertation, I can send it to you, but we do know a very significant part of that seed yield increase is a result of reducing the amount of Fusarium wilt or severity of Fusarium wilt. We do know that the calcium that is incorporated in as part of the calcium carbonate amendment also helps improve plant growth. So there's multiple factors. It's not simply just amount of reduction in the amount of Fusarium wilt. There's other benefits going on. So yes, your question is correct. It's more than just reducing the amount of disease, but in this case, the amount of disease reduction was the primary cause of yield loss. Okay, hopefully that answers your question, and if we can talk about it further, and I'd be happy to send you the work from Emily's project if you want more information. Okay, thank you. So let's talk about irrigation practices, and this can be remarkably impactful in terms of disease management, depending on the kind of climate you live in and grow in. So the whole theory behind this is, can you reduce the duration that the canopy of your crop remains wet? Can you reduce the amount of splash dispersal? Can you reduce the relative humidity in your canopy? For those of us who live in the western part of the U.S., we are blessed with typically a lower humidity, which means the ambient humidity normally is favorable for helping manage diseases. But if we're irrigating, that changes the situation. If we're growing crops that have a very dense canopy, that starts to change the situation. How can we take advantage of the naturally low humidity in a region like this and minimize the risk of spreading diseases through splashing water and so on? Most fungi and most bacteria are very readily dispersed through these means, such as extended periods of leaf wetness, splashing, and high humidity. Obviously, one of the most distinct ways is to look at a form of irrigation that reduces the ability for the canopy to get wet, and that's drip or furrow irrigation instead of using a form of overhead irrigation. If you use sprinklers, if you use center pivots, you use wheel lines, whatever it might be, anytime you wet the canopy and you create these extended periods of leaf wetness or canopy wetness, you increase the risk of certain foliar diseases, particularly bacteria and fungi. And that is, in some cases, a reason why in some areas growers have switched to drip or furrow. Of course, that's not always feasible logistically based on the type of shape of your field or slope of your field. Many people don't want to invest in the cost of drip irrigation or the technical side of drip irrigation, but it can be very, very effective. So this aspect of the economics and practicality sometimes don't always make this recommendation the most feasible. Another important aspect of irrigation that we sometimes forget is timing of irrigation. And when I say timing, I mean the the time of the day. If you irrigate first thing in the morning, so you're done irrigating perhaps by mid-morning or midday, you'll still have hours of daylight where that any moisture on the canopy can evaporate, and you reduce the extended period that the canopy remains wet, because the longer the canopy remains wet, the higher the risk of certain diseases. This, again, is not always feasible if you have a limited amount of irrigation systems and so many fields you have to get through. Sometimes it's easier to put this on a piece of paper than it is to put into practice in reality. But this can make a big difference. Excuse me, get to my next slide. So I'm going to give you an illustration here. I've worked a lot with bacterial disease of carrots, bacterial blight of carrots caused by Xanthomonas campestris carotee, also now known as Xanthomonas hortorum carotee. And we did a survey of carrot seed production in Central Oregon, also in Central Washington, over a couple of years, and I'm just going to show you a subset of this data. So in the field, O stands for Oregon. These were Oregon fields we surveyed on the x-axis, and you can see it was field K with overhead irrigation versus a part of the field that had drip irrigation. Field L that was under overhead irrigation versus a part of the field that was under drip irrigation. So we had these paired sections of the same field that were overhead versus drip irrigated. And you can see that the y-axis is the incidence of 20 plant samples that were infected with Xanthomonas at these different intervals in April, June, and August. Seed crops are usually harvested in August to September. And in 3 of these 4 combinations or paired sections of the fields, you can see that the amount of Xanthomonas at the end of season in the drip irrigated part of the field was significantly lower than in the overhead irrigated part of the field. Unfortunately, field K throws that awry, and the opposite situation was true. So it's not always a foolproof way of reducing disease pressure, but certainly in Central Oregon, more than 50% now of the carrot seed crops produced in that region are done with drip irrigation to help reduce pressure from Xanthomonas, because it is such an endemic pathogen in that area, and it is seed-borne, so it's really important to try and keep the disease level down. Okay, any questions on irrigation? It seems somewhat straightforward, but then there's always the practical reality of actually having to irrigate your crop and all that goes with it. Some other practices we've mentioned before, now I'll go into a little bit more detail on planting practices and canopy ventilation practices. So let's look at planting practices. For example, you can select the planting date to try and escape inoculum. And again, this only works under some circumstances and some crops and certain diseases. For example, you might try to avoid or minimize the risk of exposure to insect-vectored pathogens. One example for this is those of you who live in more northern areas that have colder winters. It takes a while for the spring to warm up, and certain insects don't survive in these cold winter areas. And it takes a while in the spring before the insects move from the southern states or provinces up into northern areas where it takes, warms up later in the spring. And if you can get your planting done prior to the arrival of these insects that are vectoring certain pathogens, that can be very helpful. Leaf hoppers are a good example. And besides insect vectors, also planting early to avoid pathogens that might not overwinter in your region. Selecting a planting date that's where the conditions are unfavorable for the pathogen but still favorable for the crop. This can be difficult. You may not have those particular combinations, but this is something else to think about. And then looking at your row spacing and plant spacing and your row orientation. And this ties quite a bit into the next cultural practices, which is crop ventilation practices. Your row spacing, obviously the wider the row spacing, the more likely you are to have adequate air movement through the rows. And air movement through the canopy means reduced humidity. Reduced humidity means less risk of bacterial and fungal pathogens. And likewise, plant spacing in the rows can make a difference. Now, sometimes a grower has very little choice on row spacing because the equipment may be set up with a particular row spacing based on the tractors, the distance between the wheels and the tractors and so on. But one question or aspect of planting practices that really I've seen make a tremendous difference in some growers' fields is the direction in which the rows are oriented. If you in your area of production tend to have a predominant wind direction, and if you have the practical logistical choice of redirecting your rows into that predominant wind direction, it can make a huge difference in the disease pressure of certain foliar pathogens, and I'll give you an illustration to show regarding this. And this comes into that whole aspect of ventilation practices. Thinning the canopy if necessary. If you've got too dense a population, that can reduce airflow and increase humidity, and managing your canopy for maximum air circulation. So let's look at a few examples. This is a photo I took in a buckwheat seed crop in central Washington. I was contacted by the buckwheat seed company saying they had a problem showing up in their buckwheat seed crops. They weren't sure what it was. The plants were dying back. And in central Washington, the buckwheat tends to get planted a little later in the spring because of the crops that they tend to follow in rotation, like after pea harvest. And unfortunately, this disease turned out to be beet curly-top virus, BCTV, which is carried by leafhoppers, the beet leafhopper. And beet leafhopper populations in central Washington really spike after temperatures start to warm up in the spring. So by utilizing this practice of planting buckwheat following pea harvest, it was late enough in the season that the leafhopper vector population was basically at a peak when the buckwheat plants were growing in these seed crops, and really increased the risk of these crops becoming infected with beet curly-top virus. This photo, two photos I have here are of brassica seed crops. These are cabbage seed crop trials I did here at our research center on farm and also in grower cooperative fields of brassica seed crops. We were working on two diseases, white mold caused by Sclerotinia sclerotiorum and Alternaria black spot caused by Alternaria brassicae and Alternaria brassicola. And I had just started this job, so I was trying to make sure I did a really good trial and show the growers that we knew how to produce these crops. And I had my crew do this incredibly wonderful job of staking and tying up these cabbage seed crops. And I happened to plant, just coincidentally, because I wasn't familiar with the area, we happened to plant the rows in the direction of the primary wind direction. So we had really good airflow, and we staked and tied like crazy, so we had wonderful ventilation through our rows. And despite the fact that I inoculated this trial 3 different times with the pathogen at a very high dose, we got disease, but not nearly as much disease as in the grower's field. This is down in the lower right picture, where the row direction was perpendicular to the predominant wind direction. It was a more leggy, bulky type of crop where they didn't stake and tie as well, so there was very limited airflow through the canopy. And the grower's field, even though we did not inoculate, had 3 times as much disease as our trial on station. And this was just a really healthy reminder for me how effective cultural practices can be in managing diseases. It was quite remarkable. The 2 sites were less than a quarter mile apart, same parent line, but very different canopy management, and made a huge difference in the amount of disease that developed for Alternaria black spot. And this is a picture of the disease and what it can do in brassicas. You can see what it can do to the pods. The infection can get on the seed and have a very big impact on seed germination and, of course, seed infection. Yeah, the pathogen is growing out on some brassica seed that we plated out, and there's the spores of the fungus. So definitely did result in seed infection. And we had a very strong correlation between seed germination and seed rot and the amount of Alternaria in the canopy just before harvest. Okay, so let's look at the next cultural practice, which is transplanting and hygiene. One of the aspects of transplanting that is important is to try and avoid injury to the plants during transplanting, or injury to your crop during staking and tying and other cultivation practices. Injury tends to create wounds. Wounds tend to open up opportunity for infection. It used to be a common practice with transplanting that transplants were sometimes dipped in water. That can be a big no-no for the reason of spreading pathogens like bacteria and fungi in that water. And some pathogens can be spread mechanically very, very easily, and this can be a difficulty when you're handling a lot of plant materials such as transplanting. For example, late blight in celery caused by Septoria apiicola can very readily spread on hands when people are transplanting. In cabbage, the black rot pathogen, Xanthomonas, campestris, pathspot campestris, is very, very easily spread when you're handling plants, whether it be a transplanting or in the case of seed crops, when you're staking and tying those plants. It's very, very easy to spread the pathogen, particularly if the canopy is wet. One that's quite fascinating from the perspective of those of you who either are smokers or work with smokers on your farm. Cigarettes can carry tobacco mosaic virus. Tobacco mosaic virus survives the process of drying tobacco leaves and even the process of burning tobacco leaves, and can very readily get onto the hands of smokers. And then if they then go back into the field and handle pollinaceous crops like tomato or peppers or potatoes and so on, they could spread TMV on their hands. And so that's why in many greenhouse operations, workers are required to wash their hands with soapy water once they take a break, particularly if they're smokers. Okay, so the last cultural practice we had listed overall was looking at the geographic or regional location where seed crop production occurs. And this can be on a large continental type scale where seed crops are grown in environments that are unfavorable for disease. For example, almost all the bean seed production in the U.S. occurs in places like Idaho, California, and central Washington as well, in order to minimize the risk of bacterial blights and anthracnose holototricum, because these are very readily spread by splashing water. Most pea seed production occurs in places like Idaho, Washington, and California for the same reason, to avoid this bacterial pathogen Pseudomonas, as well as Ascochyta and some other pathogens. Brassica seed is almost all produced in western Washington and western Oregon to minimize the risk of blackleg, of fungus, as well as black rot, which is Xanthomonas, because we tend to have dry summers from usually July, August, September are dry, which is right when the seed is maturing in the pods and harvested. And on a local scale, one of the things you can think about is trying to, for example, avoid areas of the farm that might be prone to frost pockets or prone to fogs or dews alongside rivers where you get a lot of fogs build up over the river. And then a very important aspect is spatial and temporal isolation of crops. And by temporal isolation is isolation in time. This becomes really important when you're dealing with biennial seed crops, where you've got an overlapping season from season to season, and that can, as we've discussed before, result in a green bridge effect for diseases. Here's an example to illustrate this, again, looking back at this pathogen Xanthomonas, campestris carotee, or Xanthomonas hortorum carotee in carrot seed crops. Here you can see symptoms on the leaves of a carrot seed crop and symptoms on a carrot umbel that was severely infected with Xanthomonas. And one of the things we did during our survey of seed crops to see how widespread it is and how the pathogen is being dispersed is we drove behind some combines that were combining these carrot seed crops. And we drove downwind at increasing distances away from these combines. You can see the dust coming out the back of these combines. These are very dry, hot climates and types of conditions in which the crops are being combined. And at the time we were driving on the back of the pickup truck, we had this piece of equipment that basically sucked the air through the vacuum and then passed the air onto these Petri plates that had a medium that allowed us to quantify how much Xanthomonas was present. And we were able to trap Xanthomonas in the air, in these aerosolized residues, a mile downwind of a combine, demonstrating how far under these conditions this pathogen potentially could be moved. And even if you do require pollen isolation distances of half a mile or a mile, you can see the potential for this pathogen to spread under those biennial systems where the new season's crop would have been planted 3 or 4 weeks ago at the time you're harvesting this crop. A really good illustration of this problem with the green bridge effect. Okay, let's move on to the topic of chemical applications and seed treatments in disease management.
Just before you do, Lindsey,
Sure.
we just have
a
question.
Take a break?
Well,
okay.
We just have a question at the bottom, and I don't know if this is a good time to address it or not.
Oh, sure. Thank you. Sorry. I appreciate you bringing that to my attention. Sorry, Lisa Ann. Do you have suggestions to avoid disease for biennial crops grown in greenhouses where rotation is an issue and getting rid of residue of annual crops? Good question, Lisa Ann. Really, in this case, residue of annual crops. I guess my questions I would ask you now is what types of crops are you dealing with? If the softer the tissue is and the more likely it is to break down in the soil, the less of an issue it might be if you incorporate those residues into the ground. I'm assuming in this greenhouse you've got bare ground as opposed to concrete. Anytime you can get rid of residues that you think might be infested with a pathogen because the crop had symptoms, it's going to be very valuable for helping reduce that likelihood that pathogen can survive to when you go into biennial crops. This is becoming an increasing problem where you see the increased use of things like hoop houses or greenhouses on bare ground where people don't have that choice of moving. And I would say one important aspect there is to not incorporate those residues into the soil, but get them out of that hoop house as much as possible so that you aren't introducing the pathogen back into the soil. I'm not sure if that adequately addresses your question, Lisa Ann. We'll wait and see your next comment. Would transplanting seed crops into pots and bringing them into greenhouse work? I think certainly you could. If logistically, practically, you can manage that amount of labor, I think you certainly could. It would make it easier for getting the material out of there afterwards. Most of these diseases, a big question to ask yourself is what diseases are you concerned about, and are they foliar diseases or truly soil-borne diseases that have a likelihood of persisting for a long time? If you're dealing with some soil-borne pathogens like white mold or verticillium that can persist in the soil for a very long time, then it may be very valuable to consider going to that extent of transplanting into pots and bringing the pots into the greenhouse. But with foliar pathogens that don't really persist in soil very long, you might not need to go quite to that extreme. Okay, thanks, Lisa Ann. And George, we now have a question coming from George.
And Lindsey, maybe you can continue with the chemical applications and seed treatments bit, and we can
Sure.
address George's question right after you finish this.
Okay, certainly. So with chemical applications and seed treatments, it's really important, whether it's organic or not, in terms of the products you're working with, is to understand the consistency and performance of these products, particularly when it comes to some of these organic materials where they're very susceptible to the kinds of conditions under which you apply them, the niche environments in which they work, the target pathogens that they're meant to be used against, and the potential for even things like phytotoxicity if they're not used correctly. There are a lot of natural plant products being promoted and utilized by growers in the organic seed industry, whether they be oils or plant extracts, compost teas. And one of the complications behind this is coming up with recommendations when there's often a lack or very limited independent data on reliability and consistency of performance. And in my program, we've probably spent at least $250,000 over the years trying to evaluate a wide range of certified organic products to see how well they might work in organic production. And it's very sad to say that the majority of the products that we've tested have not held up to the labels, the claims made on the labels. And I don't mean to put down organic products. We all would like to find some very, very effective certified organic products. But there are a lot of people almost abusing the situation that it's easy to get these biological types products registered and approved for use. You don't have to go through quite the same rigor of registration. And you can make a claim on a label without having any data to back that claim about management of certain diseases. And this is an ongoing issue as a public plant pathologist when growers ask for recommendations on how to deal with these kinds of problems. Okay, and then I wanted to mention briefly there are some products that act in a sense like vaccinations in humans, that you apply them and they trigger a resistance response in the plant as if they had been infected, and that can sometimes reduce the amount of disease if the true pathogen did arise. But I'm not quite sure. Again, we've looked at some of these, some of them are very, very expensive. Not all of them are registered for use in certified organic production, and it's question about just how viable economically and otherwise some of these are for agriculture. Okay, so George's question is, thanks for this great opportunity. We're having a small garden in the backyard, but my red cabbage is being eaten by the blackbirds, and the grapes are being eaten by the big black bug. Just want to have an explanation about these birds and those black bugs. What might help, George, is if you could perhaps take a picture of the birds and a picture of the black bugs and send them in. You could perhaps email them to me, and I can work with our entomologist to see just what those black bugs are that might be causing a problem, and we could maybe see what help we can get you. So why don't you, if you have the ability to take pictures and send them by email, we'll see if we can get you some direct aid on that question. Getting back to the slide about IPM, Integrated Pest Management, this is really important even if you're dealing with certified organic products, because it helps you look at the appropriate timing of applications to get maximum efficacy. I've many times talked with a grower who's been struggling to manage a disease, and when we started looking at the grower's management practice, it turned out that they were perhaps using an appropriate product but putting it on at completely the wrong time and the wrong application. Also getting good coverage of crops that have very dense canopies, and many seed crops have very dense canopies. And classic cases in some of these bigger, very large-scale seed crops where growers put on their applications with an aeroplane, for example, that can only put on a limited amount of water versus putting it on the spray boom, so you can get good coverage. It can make a huge difference in whether or not you're wasting your money on that application. And then another very important aspect is getting an accurate diagnosis before you try a treatment that's chemically based, because I've run into many examples over the years of people treating a disease that turned out not to be the disease they thought it was, and treatment was completely inappropriate because the diagnosis was not correct. I give you a few illustrations. This is the data we had from a cabbage seed crop. We were looking at fungicides for managing this disease called white mold. You can see the picture in the slide. And the two products that I have circled in green here are certified organic products, as well as a range of conventional fungicides. And you can see the amount of disease in the control plots was over 50%, and there's a wide range in reduction of white mold based on the efficacy of the different products, but some of the organic products also look quite good. Okay, so we have two questions that have come up. Lisa Ann asked, do you have favorite seed crop disease diagnosis books and publications to recommend? And yes, I certainly do, Lisa Ann. Perhaps maybe this is a question we can leave for the end, because that's a pretty long question to start going through. So if you don't mind, I'll wait till the end to answer that. And then Lynn Howe said, Lynn Howe, do you have suggestions for treatments that are safe for aquaponics, specifically black heart and celery and black heart and brassica? I think, again, black rot and brassica, this is a very difficult one. And my recommendation for black rot and brassicas is that
if you'd like, Lindsey, to tackle both these questions later on, if they're longer answers, we can totally do that.
Sure. Let's do that, if you don't mind. Not to put off the question completely. Let's hold off to the end, because
absolutely.
It may take a little while to go through that. And then Joanne Henderson is typing a question, but I'll keep going until her question comes up. Thank you, Albert.
No problem.
Okay. One of the things we also discovered with this particular disease that I want to mention is that there were two very critical periods of timing of fungicide applications looking at these products, whether they be conventional or organic. And this is very important to get the most out of these applications. One was early spring, when the fungus would release what's called an ascospore. That's a spore that's sprayed by the wind in early spring. And the other was late spring, when the petals from the flowering seed crop would start to fall and provide a nutrient base for the fungus. So in order to minimize the number of applications, it's really important to understand when the crop is at most risk. Okay. Here's an illustration from carrot seed crop trials we did at our research center. Some of the crew in my program doing these trials with bacterial blight. You can see again the disease affecting the umbels. And we looked at a wide range of treatments, including some organic treatments, to see if we could control Xanthomonas. On the x-axis are the different products we tested. On the y-axis is the amount of Xanthomonas per gram of seed. This is on a log scale, so the 6 means 1 million colonies per gram of seed. You can see how high the population can be on the seed. And the most effective treatments were basically, unfortunately, conventional copper type of application. And the most effective treatment was when we put it on through the sprinkler. So instead of by spray broom, we put it on by sprinkler because it resulted in carrying that product with the water all the way down the canopy to get much more effective control. So coverage can be really, really important. Okay, so we'll quickly move on to seed treatments. And if you need to treat seed because your seed is infected, we tend to look at three main ways of treating seed: physical treatment of the seed, for example, hot water, chemical treatment of the seed, and biological treatment of the seed. Let's get into a few examples. So, for example, we were looking at spinach seed treatment infected with Cladosporium and Stymphalium, and looking at hot water treatment. And you can see in the middle two figures, from left to right, the amount of Cladosporium on the seed without hot water was about 20, almost 30%. And it didn't matter which temperature we used, from 40 to 60 degrees Celsius, whether it was from 10 minutes to 40 minutes, we completely eradicated Cladosporium from the seed. This fungus, Cladosporium, is not heat tolerant, and hot water treatment can be very, very effective at cleaning up the seed. However, if you look at Stymphalium, we either could not clean it up at all, or it took very high treatments that were too harsh on the seed and actually killed the seed. So we learned very quickly that hot water treatment has a limited ability to clean up seed of Stymphalium, but is very, very effective against Cladosporium. And I'll just move quickly on from there. Another example is looking at verticillium in spinach seed. Again, we tested a wide range of products. I won't go into the details of these, but on the y-axis is the percentage seed infected with verticillium. It was over 60% in the control seed. And you can see I placed these in order of least effective to most effective, and a wide range in efficacy of these treatments for cleaning up seed. All the treatments as the gray bars are certified organic, or potentially could be certified for organic use. Okay, we also did a bunch of seed treatment trials looking at damping off in organic pea production in central Washington. A student of ours spent a couple of years looking at trials with Pythium and Rhizoctonia, and you can see again a wide range of products she tested, and this is the stand. So the higher the bar, the better in this case. This is the amount of peas that emerged. And what was interesting in her case, the only treatments that looked particularly good were Nordox, which is the organic copper product on the seed, or priming the seed, where you partially imbibe the seed before you planted. It gets the seed germinating very quickly and out of the soil before these soil-borne pathogens, Rhizoctonia and Pythium, could cause losses. Okay. So in disease resistance, I've mentioned before that this can be very, very effective if you have resistant cultivars to a disease, but may not be available. You may be growing varieties that don't have resistance. And in seed crops, often you may not even, if you're growing for a company and that's proprietary material, or you're growing a hybrid with two different parent lines, sometimes one may be very susceptible, one may be partially resistant. And this is not always an optional for a seed grower to be able to select resistant cultivars. But it's really important to understand the susceptibility of the cultivars you're growing to see what the risk might be, what management tools will be needed. And I really have this conversation very frequently with our seed growers who don't necessarily understand the susceptibility of the varieties they're given, is to communicate with the seed companies that give them contracts to learn as much as they can about the susceptibility of the parent lines they're asked to grow. An example we have is cucumber mosaic virus in spinach seed production. It turns out that the two serogroups of this virus, one of them is very readily seed-borne and seed-transmitted. And both male and female flowers can be sources of inoculum. It can be pollen-borne. It's very, very important to clean out, rogue out infected plants in the seed crops and cleaning up the stock seed. And here's the examples of the symptoms that it can cause in mature plants and in seedlings. And we showed that certain parent lines were very, very susceptible and resulted in a high rate of seed transmission. We've done some screening, for example, for resistance to Stymphalium leaf spot. Here's a wide range of spinach lines we looked at, and you can see lines on the left side are very resistant and lines on the right side are very susceptible. So you can see a wide range in susceptibility. A very, very key example for us in this region of the U.S. is Spinach Fusarium wilt. This is a picture of a hybrid spinach seed crop, and you can see all the brown rows are the female line that was dying as a result of Fusarium wilt in this field. And the male line looks pretty good because it had some fairly good resistance. And one of the things we've developed to help our spinach seed growers in this area, because they have to rotate for at least 10 years to minimize the risk, and sometimes that's not long enough. Every winter, we offer a soil bioassay. Growers bring us 5 gallons of soil from their field, and we plant out 3 spinach lines ranging from very susceptible to partially resistant. We grow it out in the greenhouse, and they can come and look at their soil. Here's an example of a soil that doesn't matter whether you had a resistant or moderate or susceptible female, everything died. It was very, very high risk. Here's an example of a moderate risk soil. So this grower knew that if they were given a contract for something that had some resistance, they may get a successful crop. If they had a susceptible line, they would have no crop. And here's an example of a soil that was completely safe, even with a highly susceptible female. And this, we've tested over 200 fields in this area for growers over the last 5 years to help them assess how safe it is to go back to spinach seed crop based on the susceptibility of the contracts they may be given. And this is just a quick summary of 2 of the years that we did this bioassay, where you can see the soils lined up on the x-axis and the risk of Fusarium wilt on the y-axis, where 1 means everything's dead. And the green bars are the susceptible female, the yellow bars are the moderate female, and the red bars, sorry, the other way, the red bars are the susceptible female, the yellow is the moderate, and the green is the partially resistant. And you can see that susceptibility of that female line really impacted how much disease develops in those fields. And so we've been able to help growers avoid these wipeouts to this disease. So kind of wrapping up on this overall aspect of disease management, it's really important when you are dealing with diseases in your seed crops to get an accurate identification, because that will determine how effective your management program might be. Monitoring the appearance and development. When does it show up? How quickly does it develop? Trying to determine, are there thresholds that for this disease that you can tolerate a certain amount or not. Roguing out symptomatic plants as soon as possible, particularly once you have an accurate identification if you know it's a very risky type of disease. It can help you assess the need for control. It can help you assess how effective your actions have been that you've implemented in the past, and help you develop a more effective disease management tool overall. So it's really important as seed growers to be aware of what are the diseases that are most common in our area, and to understand historically important and potentially new diseases in this area. And I'm going to wrap up this presentation with a quick cover of a disease that we unfortunately are seeing showing up again in the Pacific Northwest after a long period of not having to deal with this disease, and that's brassica blackleg. In the 1970s, this disease showed up in the Midwest and East Coast in states that had bought their seed from the Pacific Northwest. It was all tied to seedlots grown in western Washington and western Oregon. It was very devastating for the brassica seed industry. Some researchers, including my predecessor, did a lot of work to help resolve this and figure out how to control this disease. And it's largely been off the radar for a long time, except for the seed industry that's always been aware of the losses of the past. Unfortunately, in 2014, there was a very widespread epidemic of blackleg in crucifer crops across the Willamette Valley of Oregon. This included organic crops, conventional crops, processing, cover crops, canola, seed crops, a very wide range, and really was troubling because this disease had largely been off the radar as far as impact in this area. And here's just a quick summary showing all the counties it was found in and the very wide range of brassica crops, including weed species, on which Cynthia Oakham, who's a professor in plant pathology at Oregon State University, found this disease. Here's a photo from an overwintering organic kale crop in the Willamette Valley. It was just devastated by blackleg. Really, really troubling because this disease had not been present on any significant scale for a long time. And here, Cindy Oakham on the left in the brown jacket, and one of my students, Shannon, in the green jacket, standing on a canola crop that doesn't look too bad, but when you start to inspect the leaves underneath that canopy, there's all the blackleg lesions all over. And when we looked at a field immediately adjacent to this that had canola the season before, here the canola residues are. You can see a stem piece with these little black fruiting structures, and this is the overwintering structure where the fungus goes through sexual recombination and releases these airborne spores in the spring to infect other crops. So we know that this disease is now spreading through the Willamette Valley. Why it happened? We believe this is a case of ignorance is bliss, lack of awareness of the risks and the potential for certain diseases to cause a big impact. In the Willamette Valley, there used to be laws in place that help protect against blackleg. All crucifer seedlots used to have to be tested and treated for blackleg. Seed crops had to be pinned. There were all these rules to help protect against the risk of this disease. And I won't go into detail, but this slide basically covers some of those rules. And it included radish, which is also susceptible to blackleg. Now, some of you may be aware in Oregon, there was a big controversy about allowing canola production in the Willamette Valley. The specialty seed growers did not want canola to come in because they were concerned about risks such as blackleg as well as cross-pollination. But this bill, House bill, was implemented in about 2009 to facilitate testing canola in this region. And for some reason, at the time that they implemented this rule, they eliminated the requirement for blackleg testing of crucifer seed. At the same time, there was a very widespread planting of brassica cover crops during the economic recession, particularly, for example, grass seed growers. There wasn't a market anymore because people weren't building houses and not planting lawns. And so brassica cover crops were being planted on a wide scale in addition to canola being planted in areas of the Willamette Valley. And so this has brought up a big question for an area like Washington, which is very, very important for brassica production. We now have the Willamette Valley down in the lower left as a big blackleg flag now. In 2011, it was found in Barnes Ferry in canola production. And this spring, in March and April, I received samples from winter canola crops in 15 different fields in Idaho, close to the Washington border, as well as Umatilla County, Oregon, infected with blackleg. And the two primary areas of Washington that are very important for seed production for various brassicas, the one in the northwest is currently protected by a quarantine to help prevent blackleg from being introduced, but eastern Washington, up until next month, has not been protected, and now an emergency rule has been put in place to try and protect this area against the introduction of blackleg. And it really just reiterates for us that we shouldn't forget history. We should help be educated and educate each other about important diseases so that we don't end up in the situation that has just happened in the Willamette Valley of Oregon. And I certainly would be happy to provide further information if anyone has questions about the blackleg situation. And I use that just to illustrate this importance in our work with seed crops of disease management, because we are sharing the seed with quite a few other players in agriculture. So I don't know if that's a little longer than maybe you had hoped, but I'd be happy to ask questions if there's time now for more questions.
Absolutely, Lindsey. That was incredibly informative, and yeah, just thank you so much. We have the webinar room until 5:30, and I'm sure folks can drop off if they like, but it might be best to maybe start from some of the questions that were proposed earlier, and we'll go from there.
Cool.
So Lisanne, I think, was the one that we're waiting on, and that's a favorite seed crops disease diagnosis books or publications to recommend. And if you think this is easier to answer by putting together a resource list that we can post after, we can do that. But if you have quick recommendations right now, that'd be great.
Yeah, so I think I'll do that. I do have a list I've compiled in the past for some workshops I've done, and I can send that to you, and then maybe you can make that available through the website. Does that work?
Absolutely. Absolutely.
Okay. And then Lynn Howe asked about suggestions for treatments that are safe for aquaponics, specifically black rot and celery and black rot and brassica. It's a little difficult for me to answer this question right off hat without checking a few references, but my recommendation in this case would be preventative would be far more valuable than curative, particularly, for example, black rot and brassicas caused by Xanthomonas. Once it shows up in a crop, it can very readily spread if you're not careful, and there's almost nothing curative that will work against black rot and brassicas. So really, really important to make sure you start with clean seed. Do not buy seed that hasn't gone through testing for black rot and blackleg. This is sort of the mantra that we're preaching now everywhere, is if you buy brassica seed, make sure it's tested for black rot and blackleg, because you cannot afford to have those diseases introduced. Black rot in particular caused by Xanthomonas, if you're in an area where it can get quite warm, it loves heat, and it will do very well, not just in your brassica crop, but any brassica weeds you have in your area. So my recommendation with this is preventative, not don't get into the point where you have to try to be curative. So, Lynn, I'm not sure if that's a very satisfactory answer to your question, but you're dealing with aquaponics too, so you're not wanting to introduce chemicals, and I think really focusing on the preventative, and so you don't have to deal with the curative is important.
Excellent.
Joanne?
There's another one from Joanne,
Okay.
who I think has a similar concern as George. There's some aspects that she's having trouble identifying in her seed garden, in particular gall-type growths on one specific variety of peas. It doesn't affect the fresh pea harvest, but the mature pods as they dry seems to affect a decreased seed yield. She's wondering if she can send a photo or if you have any tips.
Yeah, go ahead and send a photo. I'm just wondering if it might even be an insect pest of some kind. If you're talking about a gall-type growth, there are some insects that can be problematic in peas, and if I'm not certain about it, I can work with some of our entomologists in the area to see if they have any thoughts on what it might be. And along this line of diagnosis of diseases and pests and so on, I obviously do this a lot for seed growers. We have WSU has a plant diagnostic clinic that deals with, basically, because I'm a researcher first and foremost, and I do help with diagnosis, but we do have a clinic that takes samples, and many states have a state diagnostic lab or a state university diagnostic lab, particularly if you're from an area that's nothing like the Pacific Northwest. It might not be something I'm familiar with. And so working with a diagnostic lab in your region where the personnel, the staff will have more familiarity with the common issues in your area can be a really valuable way to get an accurate diagnosis. But Joanne, you're welcome to send me a photo, and I'll let you know if I have any idea or forward it on to entomologists if I think it's an entomology problem. Okay, and then...
So we have a couple questions, one from George and another from Ashley. So George asks, what would be the best treatment? I suppose a bit of specificity would help, but maybe he's referring to between the cultural practices and the chemical and other seed treatment practices that you've suggested.
Yeah, okay.
Perhaps if there's one that you recommend among all of them. George, feel free to clarify.
Yeah, sure. I think that my philosophy on this is again, preventative. Preventative action, preventative management practices, and that's why many of these cultural practices become really, really important. My first recommendation through and through is start with clean material. Don't start behind the starting line by purchasing low quality or planting material, whether it be seedlings or seed that is not from a well-known reputable source where they have tested the seed, particularly if there's some notorious pathogens on that species. And then look at things like, make sure you're not growing the same crop on the same piece of ground year after year, that there's good rotation, that you've got good airflow through your canopy. A lot of those practices we've just mentioned, they're all about prevention, minimizing the risk of something coming into your area. Even things like going to a farm equipment sale, an auction, and buying used equipment, make sure you don't bring any soil with you on that equipment back to your farm, because if that equipment came out of a farm where a particular disease was present and there's soil on that equipment, it's a notorious way for exchanging diseases. I sometimes go to these conferences where they have seed swaps, and they encourage people to bring in seed and swap seed. And I'm all for the philosophy of sharing and swapping seed, but be aware that if someone had a disease on their farm and it's now on that seed, you want to be careful that you're not bringing that disease back to your farm. So again, some of it's common sense and some of it's awareness in order to make the most of these preventative treatments. So I don't know if that helps George's with a sort of a nebulous answer, but it's really about prevention.
Great. We've got two more. One from Ashley. She's wondering about the effectiveness of solarization to prevent anthracnose on bean crops.
Okay, so the effectiveness of solarization on anthracnose on beans. In a situation like this, you would only be using solarization if you were concerned that you knew you already had the infection and you knew that you had infected residues that were incorporated into the soil. The anthracnose pathogen on beans is not a true soil-borne pathogen. It'll only persist in the soil as long as the infected bean residues in the soil. So if you make sure that, let's say you've harvested a bean crop and you think you've had anthracnose, you want to get that residue either removed out of the field or incorporated into the soil. And when you incorporate it in the soil, you want to break it up into smaller pieces. The bigger the chunks, the slower it will break down. You want it incorporated into the soil with some moisture in the soil, so you're encouraging other microbes in the soil to break down that residue. And then the anthracnose pathogen will not survive in that soil once those residues are broken down. So that where you might consider using solarization is if you know you've got inoculum in that field from a previously infected crop and you're concerned that perhaps not all the residues have broken down, then you might consider solarization. And the effectiveness of that solarization is going to depend on just how hot you can get that soil temperature. And when we say hot, we mean really hot, because you want that heat to penetrate deep enough to the depth that those residues might have been incorporated into the soil. So it can be difficult if you're not in a climate that is very hot, and you'll need some kind of a clear plastic to cover the soil to get that heat to build up, like a greenhouse effect. But if you don't know that you've had anthracnose, it's not a soil-borne disease, so I wouldn't try to use it against anthracnose if you don't know that you've potentially already have had that disease introduced into your field.
Excellent. Thank you, Lindsey. There's one more from Lisanne. Any particular disease or tips you have to recommend for kale crops? And I assume disease is disease management.
Sure. Okay. The one concern that comes to mind is in light of our recent situation. I'm not sure where you're based, Lisanne, if you're on the West Coast or where you are, but kale being a brassica, the two big diseases that are always a concern in brassicas are blackleg and black rot. And another one that can be a big concern is clubroot. Now, we've talked a little bit about blackleg and black rot. I think it's really important because they're both seed-borne, is making sure you start with clean seed. Whatever your seed source might be, make sure it's reputable, tested seed. If you're concerned, you can ask the company, has the seed been tested for black rot and blackleg? And then following many of those recommendations we've talked about today during the presentation can be really important for those two. Yeah. Okay, so Joanne's going to send the photo. Okay.
And Lindsey, would it be all right to share your email on this chat box with the
Sure, you're welcome to share the email, and if it turns out it's an area that I may be a little less comfortable with, I might forward that to folks that I know have a specific expertise on that area, but you're welcome to share my email.
Great. So just pass that on, and we'll make sure that we put together a resource list based on some of the recommendations that you have for disease management books. Okay. Maybe in the interest of time, I'll wrap up with one question for you, and it's sort of what you mentioned as one of the cultural practices by defining a geography for a particular type of seed crop and knowing what crops can grow best in what regions. And so many of the growers that we work with are obviously trying to grow regionally adapted seed for a number of different crops that may not be well suited for these regions, but they like to build seed security for those crops. Sure. So I suppose, what are some recommendations or strategies for folks that are looking to grow seed crops here that don't necessarily do best, but are trying to adapt those crops to perform well in our regions here in Canada? Sure,
that's a really good question, and given everyone's interest in trying to do local food systems, that sometimes means if you want foods that aren't normally grown locally and you want to grow them locally, you might have to be creative. I've seen, for example, here in Western Washington, where in most summers, other than this summer, we're usually far too cold to do anything close to decent tomato production. And I've seen growers get really creative with, doing transplant production at first in greenhouses, putting these sloping root covers over that help increase the heat in the crop. There are various practices that people will utilize depending on the resources and time available to make something that maybe is not suited to this area be a little more suited. And sometimes it's important in looking at the creative ways you're using to try and grow a crop in an area that is not perhaps its most ideal. What impact those practices might have on diseases. So if, for example, you're providing a protective structure over a crop, it may be really good for helping keep rain off that crop, and rain, we know, creates leaf wetness, which encourages many bacteria and fungi. But sometimes, for example, in hoop houses, you can control certain foliar diseases, but you may end up creating more conducive conditions for others. For example, powdery mildews tend to do very well in greenhouses and hoop houses. That's one of the few fungi that doesn't actually require leaf wetness, but does require high humidity. The powdery mildews, if your humidity is high in the greenhouse or high in the hoop house, but you're not using overhead irrigation, and you know you're not getting the canopy wet, but your humidity is high, particularly if you have warm days and cool nights, you can end up with a lot of powdery mildew problems. So these are this is just one example of how you might need to think about your creative systems you're putting in place to grow crops that normally might be grown elsewhere. How might those practices change things like the microenvironment that would potentially influence diseases? I think those are important. This is where it comes back to understanding the pathogens that might be of concern to those particular crops you're growing, and also understanding where the risks are with the pathogen and with your crop. The more you know about your pathogen, the pathogens you might be concerned about, the more likely you can be proactive in that approach.
Excellent. Yeah, thank you very much, and I'm sure that was helpful for a lot of the folks on the webinar as well.
Okay.
We're at the end of our time here, but Lindsey, I just want to thank you so much for sharing with us your time and your knowledge. It was an exceptional presentation, and yeah, we're really lucky to have you on the call, and hopefully we get a chance to see you or hear from you again, because it's tons of really amazing information, and yeah, just exceptionally well put together. So thank you so much.
Thank you very much.
Just a few notes for the folks that are on the webinar. Our next webinar will be keeping seed in the public domain, where we'll be discussing seed banks and seed libraries. That's on September 23rd, 2015 at 4 p.m. Eastern Time. And again, you'll be able to find all the details at seedsecurity.ca. This webinar is recorded, and it will be online shortly after we're done, and we'll accompany that with Lindsey's contact information and a resource list for the topics and any other recommended resources that Lindsey has to share. So thanks everybody for joining us. We're looking forward to seeing you next time, and Lindsey, thanks again for such an excellent talk.
Thank you. It was a lot of fun.